Okay, so I know most of you. For those of you that don't, my background's in gene delivery, primarily for neuromuscular disorders. Although now in the Department of Neuroscience, we're moving towards the GI tract, that's the tangent. So rather than being heavy and in-depth on data necessarily, we're going to just sort of touch briefly on some of the ALS models and more some of the lessons that have come out from working in mouse models, and perhaps how best to craft therapeutic studies going forward. Right, my understanding is the end of the course is a proposal that you guys need to write. Some of you may be considering using mouse models or developing therapeutic studies for this. So some of this may be a bit too fundamental because I thought this was more of an undergraduate course than a graduate course. So I apologize if this comes off as a little bit beneath you, but nonetheless, here we go. So our ability to model ALS really starts in 1993, as you guys have been told already, with the discovery that SOD1 could be found in ALS familial cases. And so in that instance, in the paper from Foust, there were 11 mutations from 13 families, and a few months later, a second group with Foust in it also linked SOD1 to ALS. And so this obviously gave us a candidate gene which we can model. This is, of course, composite superoxide mutation. So an important question here is what is the nature of the mutation? Why is it that ALS can be caused by SOD? So between these two papers, you're seeing these mutations scattered all about throughout the protein, and it suggests that perhaps maybe a loss of function is what's happening. So historically, this is a little bit out of order, but for storytelling purposes, we can ask the next question was, if you take away SOD1, is that in fact what's causing ALS? And so the way that was done in 1996 was through a knockout of the mouse SOD1 gene. And the first evaluation of this animal more or less called it a normal animal for the first 6 months of its life. And so they actually went ahead and performed a peripheral nerve injury in the mice and showed that, in fact, the deficient animals did have a weakened repair response to the axonal injury. And in subsequent follow-up studies, which have asked the animals, or which have followed the animals longer out, have noticed that, in fact, SOD1 knockout mice have a distal axonopathy, so that the generation of their axons near where they're connected to the muscle, they can find increased muscle atrophy compared to control with increased reactive oxygen species. Nonetheless, you're not seeing major motor neuron dropout. Much of this is subclinical. These mice aren't presenting with any sort of overt phenotype, even if you're tracking them out past a year. So a laboratory mouse lives about 2 years of age. So just to give you guys a context. So what this data is saying is that it's not a lack of SOD activity that is leading to ALS. And in fact, in 1994, when ALS was really essentially modeled in a mouse, this was reemphasized and brought in the concept of toxic gain of function. So this was Mark Gurney's group, and they essentially, so first directed at the undergraduates. So we've knocked it out, right? We've knocked out SOD1 in the mouse, and nothing happened. So how would you model ALS? How do you get the mutation onto the mouse background? Okay, grad students, postdocs chime in. How do you get you've introduced a transgene. Nobody? All right, you introduce a transgene, right? So in this case, people are taking a genomic copy of human SOD1 with the native human SOD promoter, and they've introduced the G93A mutation, and then they put it onto the mouse. So they inject it into a blastocyte early stage or some sort of recombination event, and then the mouse that subsequently develops has this transgene in it. There are a couple of different ways you can do that. You can do it in a site-specific manner, you can do it in a non-site-specific manner. This is in mouse still has both of its copies of SOD1, and then somewhere else in the genome is this mutant copy. Okay. And these animals do have selective motor neuron degeneration and hallmarks of ALS pathology. So rapid progression, ubiquitinated conclusions, and particularly motor neuron dropout. This has become the workhorse of the field over the next 20 or so years, whether rightly or wrongly. There have been a series of versions of G93A animals, so are summed up in this table here. And really they differ by copy number. So when you're making a transgenic animal, for example, the concept you have to understand is that as your transgene integrates into the genome, there can be more than one copy of that site. And so you can, in this case, we're detecting by some sort of blotting technique, but probably a southern blot, that wherever this gene integrates into the mouse genome, you've got between 18 and 25 copies of it. And this is an estimation based on density on a plot. Okay. That doesn't necessarily mean all of them are transcriptionally active and all of them are making messenger RNA and protein. In fact, what's not shown on this table is you can go and evaluate messenger RNA levels and protein levels. But in this case, they're looking at SOD1 activity. So G93A mutation does not disrupt this mutase activity. But you can see that animals with more copies have higher mutase activity, and that more or less correlates with disease severity. Okay, lower copy numbers have delayed disease severity and slower progression. Holding them up. Okay, this is a summary of really the high expressing G93A mice from a review by Kevin Collett in 2008. The G93 mutation can be put on a series of backgrounds. In this case, we're talking about a piece in a black 6 background or a P6SJL, which is a commonly used strain as well. So it's broken down here in sort of clinical overt phenotype, cellular phenotype, and then a molecular phenotype. Basically, the mice will be largely normal through their first 60 years or 90 days. And somewhere around 90 days, maybe a little before, you can start to see what's called a rolling gait. So a normal mouse will walk. These guys will start to sort of saunter side to side, but still going in a forward direction. Not necessarily slower, they're not falling, they're not slipping, they're not doing anything like that. But if you study them, you can see that their gait is slightly different. And then as they advance, you can pick them up, and a normal mouse will splay its limbs out if you hold it up and suspended by the tail. Mice with neuromuscular weakness will tend to clasp in a little bit. They can't hold their legs out this far. Okay. And then you see a precipitous decline. So then what will happen generally is they won't be able to use their hind limbs. So then they become front-wheel drive mice. So they'll go around the cage, and their back legs will just sort of be out behind. Eventually, their forelimbs will fail, and then they sometimes can stay upright, but eventually they'll progress to where they can't stay upright. And so the different regulatory bodies have put that in as end stage. So mice that can't right themselves within 30 seconds are deemed end stage, and that's the artificial point of death. If you go down and you look at the presence of cells or the health of cells, the motor neurons themselves are largely pre-symptomatic or asymptomatic to this early stage. You do see, if you go out visually and look at the neuromuscular junction, changes in neuromuscular junction health, which will proceed in a retrograde transport or retrograde fashion towards the cell body, and then eventually you'll see motor neuron cell loss, between 100 and 120 days. I think this is somewhere around 50 or 60% motor neuron loss if you're looking at an end-stage animal. Astrocytes and microglia. I think this sort of activation of them starts earlier, but I don't know, maybe not. But basically what you'll see is they'll start ramping up. So they'll change their morphology, they'll increase in number, they'll start secreting a series of cytokines that can work go down and you look at molecular processes inside these cells, you can see that the progression of, or the symptomatic stages, excuse me, evidence of ALS starts accumulating very early, between 30 and 60 days. But again, it's all subclinical. Okay? And so it'll just accumulate down through end stage to where you have many of all of the hallmarks seen in human ALS tissue. Those are the G93A models. ALS has been, or SOD has been modeled over 20 times between 94 and now. Most of the lines, the ones that are commonly studied are G37, A4B, G85, G86. Those are the ones that are most commonly seen in the literature. But based on different copy number, based on different activity, you can see you can get a nice spread of phenotypic severity and onset and then progression. And it's largely from studying all these mouse models that we can get these ornate drawings of all the pathologies. And so they've been useful tools, obviously, to describe all this. So in addition to defining the pathology and the characteristics that sort of define the disease and how we can match it up to the humans, perhaps the most important role that these animal models are playing is the therapeutic development. And so quite often, what happens in these studies is someone will go back so that let's say they've got a pathway here, they've got a target here. Like let's say they study axonal transport as a motor in there or something that they think is playing a role in their phosphorylation site on that motor. And they think that they can modify that and somehow change disease progression or change ALS. So how that's typically done is knocking out something in this pathway and then crossing it onto an ALS model. So you'll start with two different strains of mice, one that has a knockout for your pathway of interest, one that has ALS, and then you'll cross them together. And that's how you can do sort of a preliminary study to identify a potential therapeutic target. There's been a lot of these types of studies, so we'll go over them sort of briefly and just look at some larger trends. So one of the first things that jumped out, if this can be read, I don't know that it can be read. So this says genotype model. So this is the novel pathway that's being investigated. This is the ALS model that has been crossed to. Most of these things are G93A minus. This says onset change percentage, survival change percentage. So one of the first things that we'll point out is the fact that SOD1, overexpression of wild type SOD1 can modify disease progression in ALS, at least in these animal models, right? So part of the caveat is we have this G93A model that's got 20 transgene copies. So is the disease coming from the fact that we've got a G93A mutation or the fact that we've got 20-fold more SOD than we should? So this animal in and of itself is normal. But when you start putting on ALS background, you start seeing increased severity. So if you take the regular G93A, you've got a 26% earlier onset change and a decrease in survival. So other points to take away is that most of these numbers, at least the survival numbers, seem to be clustering between 10 and 20% for changes. So not very big changes at all. And there's 3 pages of this table. So this has been exhaustively studied, going through all sorts of different pathways, whether it's apoptosis or neurotrophic support or neuroinflammation. But all of these tend to be clustering around that really short number. Another thing to take away from this is because these are knockout mice, generally, you're starting to modify your pathway of interest very early in that mouse's development, right? So it's not akin to a therapeutic study. It's a candidate. It will identify a candidate. But so, for example, let's look at BCL2. So here and modify apoptosis, and we can see a 15% change in survival. But you would argue, because really you're having a 20% change in disease onset, that this may not be the best therapeutic candidate, because it seems that all the change is happening prior before even the disease happens. Right? So the way that these things are measured in mice, generally a mouse weight curve will progress on and stay relatively flat for the length of the animal. The ALS weight curve will go up, and then retrospectively you can identify sort of a curvature where the animal will start to lose weight. It's that the apex of the curve is generally acknowledges the onset of the animal. And then when they reach that point that they can't right themselves anymore, that's end stage. So you get disease progression usually by subtracting one from the other. So those are the parameters that we're defining here. And so really from this, if you wanted to look at this critically, you want to find something that changes this column, but not necessarily this column. ND indicates not determined. Many of these studies aren't even looking at that critical. So we hit some of these points. So this is proof of concept. Do you have a pathway that's worth interrogating? Do you have a molecule that's worth interrogating some more? You know, can you develop a therapy from it? The other thing that these things might not be covered. So most of these are ubiquitous knockouts, right? So we're taking it away from every cell equally. Okay, so that doesn't necessarily tell you much about mechanism or site of action. You can have ideas about it, but it's not something that you know. You can go and engineer these further where you have tissue-specific knockouts or cell-specific knockouts to try to get at these sorts of questions. But in many cases, that's not the case. So if you've identified your candidate gene or your candidate pathway, then to me the logical progression is to an actual therapeutic study. And so this is quite different. Quite often these are performed in academic settings, so there are interesting selective pressures on academic scientists right now. Right? And so the way you get attention and the way you compete for scarce dollars is you get big differences. Right? You perform a study, you perform a therapeutic study, you get big differences. So give me a suggestion is how if you're designing a therapeutic study, how might you get a big difference? I'll lead you a little more. If we've got this mouse model, it's born and it's going to reach end stage in 130 days. Do you think if you're giving it the same treatment, are you going to have a bigger effect if you treat it starting at 30 days or if you treat it starting at 115 days? At 30 days, right? So you know how to go and get the big difference. It's probably in early treatment. Now what's the most relevant? Probably the 115 days, right? Or the later time point. So I would argue if you're gonna craft a therapeutic study in these animals, it's okay. You can do this. This is fine. Okay, sort of as a proof of concept for something that suggests that what you're doing has promise. But you've got to support it with the relevant stuff. You've got to come back at a time point that matters. Otherwise, you may just be fooling yourself. So you're broken that out a little bit. You want to attempt to use a delivery route that's relevant. If you can avoid intraperitoneal injections, that would be ideal. Intraspinal injections, in this case, it's not going to help a lot in patients. Right? The spinal cord is pretty big, and humans aren't able to cover it very well. And you don't know if these patients go through a spinal cord surgery or whatever. So you want to try to design your study so you're thinking about all that as you're going through it. And then, of course, you want, if you kill a bunch of mice, that's bad, but it's more acceptable than killing a bunch of humans, right? So people can be loose and fast with dose here, with doses that may not apply to humans. So you want to maybe try a range of doses, some that even if you induce some toxicity, that's going to be helpful for your and whether you have any sort of toxicity associated with your molecule at all, or biological. And then study design. This is not unique to ALS. This is across everything. So if you don't work with mouse models a lot, we talked about the fact that SOD models, or SOD mice live and die within 130 days to 140 days. But to get that 140 days, that's generally a number having to do with median survival. So 50% of your animals are dying before that, 50% of your animals are dying after that. That could be 115, that could be 110, right? And so you can, depending on if your groups are not successfully balanced, you can artificially create differences. And that's particularly prominent in some of these ALS models where you can have high degrees of variability. So what should you balance for? You should balance for sex, for gender. We know that in the G93A animals, that there's more severe disease is more rapid in males. So you either need to study all of one gender or all the other, or have them appropriately balanced. If your groups, if treatment A has all males and treatment B has all females, you may have artificially created a difference. You want to control for litters. So when breeding these animals, they'll have pups, anywhere from 6 to 12 pups, and roughly half of them will be affected. And so it's an easy impulse to just take litter A and assign it to this group, and take litter B and assign it to that group. There can be differences between different litters. Not all the parents are identical. And so really the proper way to do it is if you start with litter A, or litter 1, assign pup 1 to treatment A and 2 to treatment B, right? So you're splitting them that way while also balancing for sex. Ideally, as the experimenter, you want to be blinded, although sometimes that's hard with these guys. And then, of course, this is probably outside the purview here, but you need to understand what differences you're trying to detect, and are your group sizes significantly large enough or powered so that you can, in fact, confidently detect those differences. Okay, so we showed our first table with all the different genetic crosses that have been done, and so now here's the next table with all the different therapies that have been applied to SOD mice. This was also, I think, a 3-pager. Okay. So we've got agent. So here we're breaking the categories: antioxidants. This is a long page. The models that they're working in: velocity, G93A, gender, male, female, non-determined, dose. This is dosology, which is a word I'm not familiar with. When they started the dose, you'll notice some of these are quite early: 30, 70, 30. Here's 120, so maybe that's a pretty rigorous study. Again, our old friends onset and survival changes percentage, and of course the reference. So most of these are, a lot of these are starting with early onset, and then some of them are giving sort of non-specific, we're starting at disease onset as judged by the investigator, which is difficult to ascertain in the moment. It's generally a retrospective thing, at least if you're using body weight. And again, these tend to be clustering around the 10 to 20% range as far as having an impact on overall survival. Here's Rodizol, the only approved product for ALS patients. And what's interesting is that they were pre-treating and really only had a 10% difference. So through all this, it's very hard to move the needle in these ALS mice. The disease progression is very fast, and even the thing that even marginally works in patients doesn't have much of an effect. So So there's not in therapy development, there's not a threshold that you need to reach. The regulatory agencies, it's not like you need to take this animal and we're only going to approve something that moves survival 35%. That's not how it works. If you can prove that you are safe and you can prove that you're efficacious in some way, and you have enough money, you can have yourself a clinical trial. Okay, so many of these things, despite their modest effects, have gone through this next stage, which is clinical development. So this is a case in which we're starting with small molecules. My background is biologics, so the numbers here are slightly different. There may be some combination of one or two, but all this is regulated by the Food and Drug Administration. So basically, Dr. Kaspar talked with you earlier about things like high throughput screens, which are generally occurring in vitro and with assays, something like a cell culture. So you're just dumping a bunch of things on and screening them as fast and as efficiently as you can, looking for a quick readout, a yes, no readout. And then you might get some leads and you might take them to animal testing. Okay. And then from your animal testing, some candidates may emerge. You can go through some optimization and the like. But ultimately, you're going to get the phase 1 clinical trial. And this is a small group of patients. Depending on the circumstance, you could be working in healthy volunteers. For rare diseases, you may be working in patients, actual patients. But the sole purpose of phase 1 is to prove that your treatment is safe. So you'll start, usually you'll have 3 or 4 different dose cohorts. They escalate, they'll be spaced out by a period of time so that you can dose a series of people with low dose, have a break, evaluate them if you didn't kill them or hurt them in any way, and then you'll move on to a medium dose and then a higher dose. Ultimately, you should be ending on what you think is an efficacious dose based on what your earlier predicted, or on your preclinical studies. I'm not sure whether they're getting 40 to 60% success rate. Phase 1s are generally not blinded. My understanding is if you failed in phase 1, you've done something really wrong. Things tend to work in phase 1. But this is a slow process. So here we're already showing 7, 8.5 years, and between $1 and $5 million, just to know if your drug is safe, and you might not even be into your candidate population. Phase 2, you start to ask if your stuff is working at all. And so you'll start to recruit volunteers. So phase 1, at least for biologics, so I can't question these numbers for small molecules, but for biologics, this tends to be a number around 10 for rare diseases, biologics. Phase 2, you're asking efficacy, so you're going to be in patients, and you're probably somewhere up to 30 patients or so, between 10 and 30 patients, biologics, and rare diseases. $5 to $20 million. So academia and small operations can generally go through phase 1. From here on, you're going to partner with somebody. So that's your advisors and your work. So they're the ones that will keep the pocket. And so a good deal of ALS compounds have gone through many of these stages and are not showing defined evidence, including IGF, which is the subject of 3 trials with mixed results, and even making it through phase 3 without any conclusion. So more tables. This is from a review in the Lancet from trials from 2004 to 2014. Many of these are the same molecules that were tested in mice. They went on the trial with really minimal or no effects. So some of these are evaluating survival. Some of these are evaluating isometric contractions. Some of these are evaluating functional rating scales. And they're not moving the needles in these patients. So why haven't there been any successes? Any ideas? You were supposed to give an answer.
If you look at patients in the clinical trial setting, some of them the disease has progressed too far that there really isn't anything you can do.
patient that we're enrolling is onset of 6 months ago, maybe our data, did we actually run that test in the animal? You don't know, probably not, right? So that's possible. That can go under the broad heading of misleading preclinical studies. Even has to be studies where they were dosing too early, or not dosing late enough necessarily. It could be patient heterogeneity, right? We're testing an SOD, which is 2% of patients. Most of these trials are not SOD only patients, right? They're familial, they're more patients, or whatever. Perhaps the dosing that you define in a mouse is not appropriate for a human. The best we can do is draw a straight line between, but not everything is going to work that way. So are you even testing it at an efficacious dose? Definitely a safe dose, but is that enough to work? Some of the IGF trials were giving subcutaneous delivery. IGF is not a stable molecule, doesn't pass the blood-brain barrier very well. So is that necessarily the right route to treat those patients with that molecule? Most of the studies in mice are not giving the mice growth hormone, which is almost all ALS patients are on, or what any other drugs that the patients take. So it's a fallback that a lot of people are using. Well, our mice studies weren't full of the drug interaction. Trial design methodology. So there's a debate. I think most ALS studies right now will follow patients 12 months to 18 months post-treatment. Should they be longer, should they go 24 months? That's something that clinicians would debate, touch on their patient population. Proof of biological effect. So ideally, you want to be able to measure the drug that you're giving, or the biologic that you're giving is working. And not a lot of these, first of all, we don't necessarily know the mechanism of all of the things that we think we're giving them. And importantly, the site of action that we would measure is where? It's inside the CNS, right? It's not so easy to biopsy that, figure out am I having a function inside the spinal cord. And so an important question for a lot of these things is it even, we don't know if we've tested what we think we've tested, right? We just know that we gave it to these people and it didn't work. Did it actually do what we meant it to do? You don't know. So that's sort of a problem that the animals haven't been able to predict. So let's go back and revisit misleading preclinical studies. So there's a group called the ALS Therapeutic Development Institute, and they're not the most popular group. What they have done, essentially, is gone back through all the compounds, and not all of them, but a great deal of them, the ones that have attracted most attention, the ones that have gone from the trial. And they've essentially tried to replicate the data that was in the original publication. They try to replicate it with what they feel is an appropriately designed study. So they're trying to balance the sex, trying to balance they're trying to, instead of group sizes of 7, or group sizes of 30. And they're having trouble replicating these things. So turquoise is what was originally published. Right? So we're getting here's our 10 to 30% survival that we're seeing across all those tables that we're looking at. And then here's ALS TDI trying to replicate those studies. And they can't. So they're not the most popular group because they have essentially told everybody that they don't know how to run a study, or that you don't know how to run a study. But there's probably a degree of merit to what it is that they're saying. So as a field, we need to step up how these things are performed. Another fallback argument, which may or may not have merit, is that the mouse model, the SOD mouse model, is not a good predictor. It's a bad mouse model. so people have, as we've discovered new genes that you've heard about, of course, groups have attempted to model ALS in other ways, using Foust, using 472. They're finding that challenging, certainly not as robust as the SOD models. So you touched on this in your lecture, Catherine. Could you go over this model, C9orf72? It's not a great model. It doesn't really have pathology. So they made a model.
No, I didn't specifically go into.
Okay, they have a model which they've made an inducible allele in which they put an expanded repeat in front of GFP, and then they turn it on with the TET activator. And what they can find is but importantly, they don't find signs of brand translation. They don't find signs of this being translated to protein at all. But for GFP by itself. What they can find is RNA foci with no degeneration. So we still haven't modeled C9orf72, but Foust knockouts that lead to early lethality. Here a group has put mutant Foust alleles onto a wild-type rat background. It's inducible, and they're finding changes in very, very early, very severe disease. But interestingly, you're seeing distal pathology. So if we're looking at the intact neuromuscular junction, so this is neuromuscular junction, this is the nerve coming in. The red is the receptors on the muscle that you're labeling. And so you're seeing nice overlap in the non-transgenic, but you're seeing that there's no real branching here in this particular transgenic rat. So we're finding disruptions at the neuromuscular junction, but even at end stage, at end stage, we're not finding abundant motor neuron degeneration. So it's a mystery that they don't quite know yet as far as why these things are happening, but it's not unheard of because attempts to model ALS using TP43 are running into the same thing. So more tables of exhaustive mouse models. Knockout of TP43. In this case, we're using a motor neuron specific depletion of TP43 as sort of a delayed motor neuron disease. You can see the motor neurons drop out quite profoundly, but it's not necessarily the case of what's happening in ALS patients. We're seeing point mutation. And so if you, I've gone out of order here, but basically, think back to SOD1. Wild-type SOD1 overexpression in and of itself was not harmful. That's not the case what we're finding in the TP43. So if you take wild-type human or mouse TP43 and overexpress it on a wild-type background, sometimes mildly 2 or 4-fold over, you can create some sort of early degenerative disease with motor symptoms. But interestingly, if you look at degeneration for lower motor neurons, you're seeing no, not determined, 10%, 25%. So many of these TDP43 models, whether they're wild-type overexpressors or whether they're mutant overexpressors, are not seeing hallmarks of motor neuron degeneration. Even though some of the animals have motor phenotypes, some of the animals don't. So our TDP43 models right now are still a bit of a mystery, as are the Foust models, as are the C9orf72 models. So we're kind of still working with the SOD model, for better or worse. So in trying to sum all that up, the toxicities when you're adding these transgenes over the mouse background appear to be dose-dependent. They're certainly dose-dependent. They're more predictable when you're introducing the transgene with a mutation in it. We didn't touch on this, we did touch on this actually. Gender specific. So there are important gender differences when working with these animals. And then strain specific, which we didn't touch on. But if you move your mutation to different background strains, you can get variability. So that's why it's important to balance and we live. Many of these processes are F1 processes. And then their toxicity can be influenced by the overexpression of the wild type. argue that the mouse model, at least the FD mouse model, is flawed and we need to throw it out. I don't think that's the case. I think if you go after a bona fide target, which we're able to do with this gene therapy, which we'll go over here in a second, you can see effects that we believe to be real, even in a time-relevant manner. So the tool we're going to use here is adeno-associated viral vectors. Probably the rest of this a little bit. AAV vectors AAV, it's a small non-pathogenic virus. 80% of the population is already seropositive for it. It doesn't cause a disease. There are over 150 isolates for different varieties of AAV. What this means is they've got a different protein code, so they're using different receptors, they've got different tissue tropisms. But their biology is pretty simple. They're easily turned into a gene therapy vector. There's two main open reading frames. To make AAV a vector, you remove a vector. The only thing that's left are two non-coding terminal repeats. And then you just insert your transgene set in there, and then off you go. That's relevant. I want to deal with that right now. AAV is safe. I think we're across all these different clinical trials. We're 1,000 patients or over 1,000 patients dosed with no vector-related adverse events, including patient one. These range from eye diseases to liver diseases to lung diseases to brain diseases. So it's been a series of issues. And in fact, within the past 5 or 7 years, we're starting to see efficacy in early phase clinical trials, clinically meaningful efficacy. So Leber's congenital amaurosis, which is a congenital form of blindness and retinal degeneration. Direct eye injections are literally taking blind people and being able to make them see. And then in hemophilia, generally, so that's a blood clotting deficiency. Patients are generally treated with protein replacement, periodic protein replacement, so they're taking out blood clots. The medium high-dose patients, I think all of them have experienced either a sort of extension between when they need their protein replacements or haven't needed protein replacements for past a year, past a year. So essentially making them not quite normal because you're not at 100% of your protein level, but having a clinically meaningful effect. So the blood-brain barrier for a long time has been effective at keeping out invading pathogens as gene therapy vectors. We were fortunate to discover in the gap of our lab that a particular serotype or variety of AAV could cross the blood-brain barrier following an intravenous injection in mice. And luckily for us, it hit cells that were relevant to ALS. It had early time points that hit motor neurons, at later time points it hit astrocytes, which you've learned have a heavy influence in controlling disease progression, at least in the SODN. And so this is a horizontal section through a mouse model where everywhere you see green GFP introduced by a single AAV 9 and 10. So it became real logical to us to try to diminish SOD1, mutant SOD1 expression in ALS mice, or G93A mice. And so this is basically the development of an sRNA construct, we tested 4 constructs in vitro, and looked at knockdown. All of them perform similarly. I think we selected 130 to advance to animals. And so this is tissue modernization of the spinal cord after injection of our shRNA construct. Mice injection of P1 or P21, and in the sac, I think about 30 days post-injection. And so looking at SOD expression, you can see quite a dramatic effect in P1, less so with detectable P21. So that's more like here. Those are early time points, those are preclinical time points, as you probably picked up on. But when we go and actually run rapid onset, or rapidly progressing P93A animals. So in this course, have you guys gone over the study of sort of the roles of the different cell types of motor neurons that are largely controlling onset and glia largely? Okay. So the P1 injected animals, I haven't shown you this, but the P1 injected animals are going to be targeting about 60% of the spinal nerve. Okay, so the expectation going into this study is that you would lose onset. Okay. Treatment of P21 is going to hit mostly glia, like we were showing you before on that previous picture, and as well as P85. So the prediction is that these would not influence disease onset. That's what we're showing you here. So this is an unaffected disease onset. And we see that the 21 and 85 and control guys are all clustering very well around between 90 and 120 days. But we see that we've delayed onset in the P1 animals. And then if we ask about survival, now we can start to see our 21 and 85 day guys separate from our control treated animals. And again, P1, which is a group of motor neurons, is seeing even greater change. That's quantified here. So this is our change in weight, our inflection point in the weight curve as we're defining, so we're looking at that retrospectively. And then here's our total survival, and we subtract one from the other, and we can determine the disease duration. But importantly, even if you look at our P85 time point, we're seeing a 30-day change in survival, which as a percentage is about 22%. Really is always 10. When we look at the functional recovery of these animals, or function of these animals, we see that we can, we don't have wild type ones. We see that their mass is in the early treated guys, is retained a little later, although our P85, they do diverge from the control animals a little bit. This is grip strength, not statistically significant, but possibly trending towards improvement over control. And then row performance, again, sort of the same situation. This is going to show up at all. All right, now, this is non-quantitative measurement of SFD learning, of course. This is control. This is P1 treated, this is P21 and P85. So if you can see this at all, this has the most SFD expression, this has the least SFD expression, and these guys are sort of in median. We're winding down here. If you go and you look at an individual cell level, so our vector that we're delivering, in addition to an shRNA construct, also has ESP, so we can see that the cells that we're targeting, we can identify the cells that we're targeting. So if you look in the spinal cord at P1, so we're seeing green round cells, which, depending on where we are, tells us that motor neurons, they co-label with choline-acetyltransferase and motor neuron marker, but importantly, their SFD expression is diminished. Here's a motor neuron that's not well expressing DFP, and we can see robust SFD expression in that one. Importantly, in P21, we're seeing really an absence of green motor neurons, but they're still there. Even though they're maintaining the rest of the expression the same as in P85. So we mentioned this, that's 22%, so that's a pretty large number when treating at that time point these animals. Specifically targeting the glia is how it's generating that effect. And then treatment of the P1 animals is targeting motor neurons and slowing your length of disease onset. So next we've partnered with Don Cleveland's group at California, San Diego, who has a G37R animal, which has a much later disease onset and much slower progression. And so their onset is nearly 200 days, and we treated these guys definitively after onset. The average treatment was 215 days. And our ends went up much more first group actually was untreated control. This group was a mixture of GFP and untreated control. So overall, a more rigorous study than the second model. So our onset, our groups were balanced for onset. Again, treatment is happening after 2/3 of the animals have already begun showing signs of ALS. But importantly, their total survival is significantly shifted down. So we dive into that further. Because this progression is so slowly, they've broken it out to early and late disease. So we seem to be having the most effect in early disease progression. And our total disease duration was changed by 66 days, giving us a survival difference of 86 days. Interestingly enough, this is also 22%. So we're getting the same results in 2 mouse models. And they're having moderate functional recovery on group strength tests. So what I didn't tell you is that the G37R animal is floxed, meaning it's got lots of P sites on either side of the mutant allele. If you did a preview, we can find that out and study it in tissues with the manner. So Don group had done this previously by using CRE driven by an astrocyte-specific GFAP promoter, and the improvement that they got was 60 days. So we were quite happy to see that robust result. So all these studies were intravenous, and for lots of reasons, not lots of reasons, but for a variety of reasons, intravenous is probably not what's going to be translational for patients. The fact is, what is likely translational is delivery into the cerebrospinal fluid, so lumbar puncture or into the sternal back or something like that. So that's the base of the spine or the base of the skull. That's technically challenging in mice, not very reliable. People claim they can do it. People claim they can do it. Just to finish up that study with Caspar Lab, then went and did intrathecal into non-human primates or monkeys, and followed them for a couple of weeks and asked whether they could knock down SODs. So this is a nice work from Laura Ferry Wolo. How close is that? They're close? Pretty close. All right. So basically, here you can see a myelogram in which they've injected virus into the lumbar spinal cord. You can see the white ghosting up and down the spinal cord, or excuse me, the lumbar space, intrathecal space, and you can see that it's migrating up the spinal cord. What we're looking is die. Virus itself doesn't show up on the scanner. And so we're using our same construct, and then we've got GFP and we've got our SOD, shRNA, and this time directed to non-human primates. And so we're seeing black is GFP immunostaining, and we're seeing robust induction throughout at least the ventral aspect of the spinal cord. And then when you capture regions of the spinal cord, specifically for and measure SOD levels, these aren't transgenic functions. There's not a lot of transgenic functions right now. This is just measuring wild type SOD knockdown. Okay, so whereas the mice were starting with 20-fold overexpression, here we're just starting with wild type. And what Laura is showing is that she's knocking down nearly 90% of SOD expression. And it's occurring in motor neurons. She did some laser capture work where she selectively looked at motor neurons, and then she selectively looked at surrounding neuropil, which is going to be mostly capturing glial cells. As you can see, nearly 50% knockdown there. So, right, yes, multiple levels of the spinal cord. And so this quantification was occurring at the level of the lumbar spinal cord, but importantly, when we give that single injection, we're getting up through the cervical thoracic region as well, and seeing RNA levels decrease at all 3 places. So Brian's group, in conjunction with Don, are trying to translate that to clinical trial. rigor, do a couple models,